hello, and a warm welcome to the fifth session of our new speaker series, A Sense of Place, the Human Connection to Earth, Water, and Sky. It is also our 26th session of the overall series. And to all of you that have come in, we're having a little problem with some of the IT connections, and so we think probably the coyotes at work, but Chris is going to overcome it, and we will continue. This November, we are celebrating Native American Heritage Month. It's a strong reminder of the destructive effects of colonialism that still remain today. It is also a reminder that we have survived, and as our speaker, Ngone Blair, has said, this is her quote, as First Peoples, we come from this land and we honor our relationships and responsibilities to the land, water, plants, and animals who are our original instructors. We have never forgotten those teachings, and they are needed now more than ever. Thank you for that quote. I will continue on. Today, we are very honored to host Stephanie Ngonekodakwe Blair, hoping I'm saying it right, a doctoral student from the School of the Environment at Washington State University, based in Puyallup. Hold on. Okay, based in Puyallup, Washington. Her own heritage is Chippewa and Inupiaq. The title of her talk is Saving Pacific Salmon Through Chemistry, Toxicology, and Traditional Indigenous Knowledge. This series is hosted by the Indigenous Education Institute. Today's session is supported and funded by Oregon State University Pacific Northwest National Laboratory Superfund Research Center from Corvallis, Oregon. My name is Dr. Nancy Maryboy, and I am from Tsetlagai des Ah, which is White Rocks, Utah in Navajo, and I'm also from the San Juan Islands, Washington. I'm Diné and Tsalagi, that means Navajo and Cherokee. My Cherokee name is Tsawayuga, and I am from the Cherokee Bird Clan. My Diné Navajo clans are Bibatoni, the Deer Springs clan, and Senjakini, the Cliff Dweller clan, which has relationships with Santa Clara Pueblo. I'm an affiliate professor in the Department of Environmental Studies and Forestry at the University of Washington. I'm also the president and executive director of the Indigenous Education Institute, which has been in existence for 30 years. We are located in the beautiful San Juan Islands of Washington State and on the beautiful Navajo Nation. IEI, the Indigenous Education Institute, is an Indigenous institution with a mission of preserving, protecting, and applying traditional Indigenous knowledge today. We also honor our many non-Indigenous allies from around the world and work with them in dynamic and collaborative relationships. I would like to begin today with a heartfelt acknowledgement of the Indigenous people of our Mother Earth, to honor our many participants from around the world. I also want to acknowledge that I currently reside on the ancestral lands of the saltwater Salmon people of the Salish Sea, who have called this place home since time immemorial. I deeply honor the inherent and acquired treaty rights of these Indigenous peoples. The speakers and presentations in this series have been chosen to reflect an awareness of the foundations of traditional Indigenous knowing and living. We also honor and work with practitioners of Western science, bringing together two ways in a collaborative process of integrity and respect. We use the concept collaboration with integrity, which is the title of one of our books. In this series, we are focusing on the human connection with our earth, waters, and sky. A major focus for this speaker series is expanding awareness and understanding of cultural differences and similarities, and to support successful and diverse collaborative working relationships, whether it be at NASA HEAT, Johns Hopkins University Applied Physics Laboratory, the National Science Foundation, or in education, health, astronomy, national resource management, museums, science centers, national parks, and tribal communities. I want to thank you personally for attending this webinar. The interest you've shown is heartwarming. We have many people registered from all across the United States and Canada. We have participants from around the world, including Canada and South Africa, to name a few. We also have participants from 38 different tribes who have registered, and we have over 300 people in this audience today. We also are welcoming many classrooms and professors, museum personnel, and national parks personnel who have registered for this event. Now, before we continue, I would like to ask you to put any questions for the presenters into the question and answer box and not into the chat box. Okay? This will enable us to better serve you towards the end of this presentation. We are honored to have Dr. Diana Rollman, Oregon State University Superfund Research Center, to serve as our question and answer moderator. And now I would like to introduce Ngone Blair. Ngone received her Bachelor of Science in Chemistry and her Master of Environmental Studies from Evergreen State College in Olympia, Washington. She's an enrolled member of the Sault Ste. Marie tribe of Chippewa Indians in Michigan and the Nome Eskimo community in Alaska. She's currently the environmental toxicologist for the Confederated Tribes of Umatilla Environmental Sciences Program, where she is leading the development of the tribe's toxics inventory and strategic plan. She's a doctoral candidate at Washington State University's School of the Environment at the Puyallup Research and Extension Center, studying the toxic effects of urban stormwater runoff on Pacific salmon. She's studying the toxicology underlying the urban runoff mortality syndrome, a condition that kills coho salmon when they return to urban creeks to spawn. In her research to save Pacific salmon, she draws on Western science as well as the traditional knowledge and connections of her native communities. I'm having a little, let's see. She shares her research with urban Native youth, inspiring their interest in STEM, as well as involving them with projects that connect them with their own tribal heritage. And now, without further ado, I would like to present Ngone Blair.
Thank you so much, Miigwech. Nagana Kodokwe Indigenous Kaz, Makwando Dame, Ojibwe Anishinaabe Kwe Indao. Hello, everyone. My name is Nagana Kodokwe. It's my Ojibwe name. My English name is Stephanie Blair, and thank you so much for that wonderful introduction. I wanted to share this initial slide to give you just a glimpse of what you heard about my past research with coho salmon and the toxicology of urban stormwater runoff, which I'll give a little bit more of a background on our research findings. But I grew up in this place. You can see the satellite image here of the Salish Sea, and Tacoma, Washington was, a place that was both urban and surrounded by lots of water and landscapes that were fed by salmon for millennia. My family are Ojibwe, Inupiaq, and then married into many of the Coast Salish tribes, including Puyallup and Tulalip, Nisqually, Makah. So this was the place where I first became part of our tribal communities, both in our urban landscapes and extending to my many relatives and families that are from places that taught me about what it means to be in community, even though I grew up far away from my traditional homelands. And that journey of connecting to our Ojibwe and our Inupiaq identities and our ways of life was very much interwoven in the story of this place and what brought me to my current research as an environmental toxicologist. So some of the pictures that you see here are of me in the bottom right corner with my dad, who that's from our Ojibwe family, where we spent all of our time when, we weren't in the city, we were out in the mountains fishing and hunting. And so my relationship with our first foods was very much not necessarily spoken of as traditional, or this was our treaty, exercising our treaty rights, but it was just part of our way of life to be able to explore Washington State and to really connect with how beautiful this particular landscape really is. And being in the urban environment as I, got older, I very much wanted to give back to this sense of place that is very dynamic in the types of communities I've been a part of. So I feel very fortunate that my path has led me to our research lab at Washington State University, where I got to explore the complex chemical nature of our urban stormwater runoff, especially the contaminants that come from our urban roadways and infrastructures and vehicles, where we're still learning about the toxicity of the many, many chemicals that enter our estuaries and our very valuable streams and rivers and lakes in the Salish Sea. So in King County, especially in the around the city of Seattle, the story of 6-PPD quinone, which is a newly discovered chemical that our research group was a part of identifying. It came from this problem of what's called urban runoff mortality syndrome, where there were these unexplained die-off events of coho salmon in these streams near Seattle. And the research group that I was a part of that led to the discovery of 6-PPD quinone had been investigating this particular phenomenon of these coho salmon die-offs. And they also were looking at the complex nature of urban stormwater. So we know that urban stormwater pollution, all of the contaminants that come off of our roadways, our roofs, all of our parking lots, many times that just enters directly into our nearby streams without having any sort of treatment. So this in the Puget Sound is recognized as the #1 source of pollutants to the Puget Sound. And a lot of scientists from the NOAA's Northwest Fisheries Science Center to Washington State Department of Ecology formed these very interdisciplinary collaborative groups to figure out which of these pollutants were the most toxic. Now, having spent the last 8 years of my experience as a graduate student investigating this complex mixture of urban stormwater, often our introductory slides start with a figure much like this, talking about how development in our urban spaces is almost considered to be a natural phenomenon, that all of the cities that we have built and our impermeable surfaces, like our roofs and our roadways, that collect that runoff and carry with it all of those contaminants into our streams, just developed naturally. And our goal to redevelop our urban spaces to mimic the natural landscape, more like the forest that allowed more infiltration into the ground so that that stormwater can be treated through the natural filtration, is something that we're moving towards. But this narrative often ignores the presence of Coast Salish people and other Indigenous peoples that have been managing these landscapes. And so there's often this need to also re-examine our history about why we developed the spaces that we did and who was displaced and who should be involved in managing the care that's needed to address these modern problems we have with our urban landscapes. So, urban stream syndrome is a term that often describes the multivariate effects that happen in our urban landscapes that are related to water quantity issues, such as flooding, and the water quality issues that bring those toxics and those pollutants into those streams that affect aquatic biota. You see more eutrophication or more algal blooms. And that causes a whole host of issues for our salmon and other really sensitive and important keystone species. But what often is invisible are all of our chemical environments, all of the mixtures of pesticides to fertilizers to flame retardants, emerging contaminants that all accumulate on those surfaces and they enter into what would be a familiar storm drain side of a parking lot or a roadway. And this is usually all that you would see, maybe some pieces of plastic or an oil sheen. But in there is quite a complex mixture of contaminants that scientists, such as those in our research group, have been trying to understand which of those are the most toxic, but also how we have mixtures effects or how we have combined toxicities that make it a challenge to recover species like coho salmon. Now, our research group that we partnered with, also with the Center for Urban Waters, University of Washington, Tacoma, where they have a team of analytical chemists who have been looking at this chemical complexity, especially from our urban roadways and vehicle sources. And before we were able to identify 6-PPD quinone, what was an important finding from looking at that, characterizing that complexity is how little we really understand about the even identifying what those contaminants are. And even if we can identify them, usually we lack knowledge on the toxicity of the chemicals that we can identify, which can number into the thousands. From a toxicology standpoint, how we usually learn about harmful contaminants is from two main pathways or two main discovery pathways. The first being intentional screening and discovery. So we have protocols that we follow through the EPA where we have test organisms or we have laboratory-based testing with cells or tissues that we can use to look for known types of toxicity, to be able to screen new chemicals or to test for chemicals that are currently in our environments, but we're trying to understand ways that they could be harmful to our ecosystems and our human health. But often the way that we discover harm is through the real world cues. So finding anomalies in our biological systems, like the coho pre-spawn mortality phenomenon, that led investigators to focus on what contaminants were entering those urban streams. Now, fish kills are pretty uncommon these days, so the fact that, that we were able to track this contaminant because it was so highly toxic to this important species, that really allowed us to find that acute toxicity or that chemical that was driving that. But there's many forms of toxicities that don't lead to such obvious fish kills, and those are much harder for us to gain an understanding, as well as the contaminants that drive that. Now, for our work in our laboratory at WSU, we had quite a number of documentation of these field studies that looked at coho pre-spawn mortality. And when those surveys were being conducted, they also noticed a suite of symptoms that the adult coho would display before they die that were very abnormal. So this was the surfacing behavior that you're seeing in the juvenile coho salmon from our WSU laboratory, and it very closely matched what we were seeing in the streams. So this very otherwise healthy adult coho salmon on the left and a juvenile coho salmon in the lab on the right. They showed these behaviors of what looked like a healthy fish being oxygen deprived. So it's sticking their nose out of the water and gasping at the surface. And very soon after exposure to the roadway runoff in the lab and in the urban streams, these fish would die in just a matter of hours. So a very extreme response. Now, my research focused on the fish cardiovascular system based on these symptoms of oxygen deprivation. Early on, my research for my PhD was looking at the impacts to the coho physiology. So one of the first places we looked was at the blood pathophysiology, what was happening to the blood that could tell us some clues about how they could die so quickly. And one of the key pieces of evidence that we found was what is called a severe hematocrit rise, which means that the blood gets really thick. The hematocrit being defined as the volume of the blood that's taken up by those red blood cells. So what you see in this figure is a juvenile coho salmon exposed to clean water in the control group on the left. And then on the right were the coho salmon exposed to roadway runoff that we collected from the highway in Seattle at State Route 520, and then also in Tacoma, Washington. So you can see from the figure that the blood gets quite thick very quickly when those fish start developing those symptoms of that oxygen deprivation. Now, we tested a couple of different theories about what could be happening to a fish physiologically to cause that blood to get very thick. And then the one that we found positive results, or we led us down the right path to understand what was happening to the fish, were impacts to their blood-brain barrier. So this idea that in a fish's body, its vascular system shouldn't be permeable to any of that blood plasma leaking out of the blood vessels, which would cause that blood to thicken. And the two places that have these impermeable blood vessels would be both in the brain and in the gills. So for my research, we tested impacts to the permeability of the blood-brain barrier at the point of the fish developing these surfacing symptoms by exposing them to the roadway runoff until they became symptomatic, coming to the surface and sticking their nose out of the water. And then we put them under anesthesia, and then introduced these tracers into their circulation. And these tracers would go through their blood for a period of about 15 minutes. And then we would wash the blood vessels out with saline. And this would remove any of those tracers from the blood vessels in the vascular system. So anything that had leaked into the brain or leaked out of the gills, we could then measure in those tissues directly. So we focused on the brain, and after circulating and washing out those blood vessels, we then dissected the brains, and then were able to homogenize that material and then use a fluorimetry technique to measure the fluorescent tracers that we introduced into their circulatory system. So any fluorescence we detected in the brain would tell us that we have blood-brain barrier disruption. So in this picture, you can see on the left that brain is white, which means we had washed all of the tracer out of the blood vessels, no blood-brain barrier disruption. But on the right, you can see the yellowing from one of the tracers called fluorescein, where you can visually see that all of that tracer had leaked into those blood vessels. Another way that we look at pathways that are related to blood-brain barrier disruption is looking at upregulation of these proteins that are responsible for blood-brain barrier permeability at the junctions between these endothelial cells. So here is just showing you two cells that make up the blood vessel walls and some of the proteins that are involved with opening or closing those junctions like a gate. So there's a lot of signaling that can take place in response to systemic inflammation or oxygen deprivation that can control that blood-brain barrier permeability. So this is showing you more results from our studies that were really started to give us more evidence that blood-brain barrier disruption was indeed the cause for the the blood plasma leaking out of the blood vessels and causing that acute respiratory distress and the symptoms of oxygen deprivation. So we used histological techniques, and then we also used a technique called RT-qPCR to also show that there were changes in those proteins that control blood-brain barrier permeability. And this is another figure that we used to show that as the symptoms developed, we had a separation between groups of the juvenile coho that we exposed to the runoff, and we sampled them in stages, where we had a group that was exposed for 3 hours, but they were sampled and tested for blood-brain barrier disruption before developing those symptoms, at the onset of the surfacing symptoms, and at the loss of equilibrium stage, where it becomes the most severe, and we see that severe hematocrit rise. And here, what this figure is showing is that whenever you have that fluorescein showing up in that brain tissue, which is the y-axis, the vertical axis, and then the exposure time on the x-axis, we are able to see a really tight correlation between the development of those surfacing symptoms and the accumulation of that tracer in the brain, which indicates for the blood-brain barrier disruption occurring. So this was a key piece of evidence showing that the disruption was indeed very closely associated with the surfacing symptoms, and that even in just a couple of the pre-symptomatic fish, we were able to detect that fluorescence, indicating that it can occur prior to the surfacing symptoms, which may be driving the reason why we see that acute mortality response. Now, another interesting observation we made that we didn't predict, we were just focusing on the brain in the beginning, was that we also saw leaking coming out of the gills whenever we saw blood-brain barrier disruption, again showing that there is a massive loss of the blood plasma, causing the blood to thicken, causing the fish to die very quickly. But also, this is showing that we have some type of systemic response, that it's not something that is just happening in the fish's brain, but something interesting is driving the coho's vasculature to become quite permeable, really in a matter of hours, so very quickly. And we still don't quite understand what is driving these types of pathways, but it's really important from a toxicologist standpoint to understand the molecular events that drive this. And again, we're still learning from species like coho about what these molecular events might be. But having that knowledge of those pathways help us develop better tools to be able to screen for the complex chemical mixtures of roadway runoff and other chemicals that are in our environment. So research labs like ours at WSU are trying to identify what is that molecular initiating event. And the research that we've done so far looking at the blood-brain barrier has really helped us better understand what pathways we can look at in our laboratory research. So this type of work is referred to as an adverse outcome pathway. So linking together what that adverse response is that drives sudden death or coho acute mortality, and trying to look at what those domino effects might be at the beginning that could be initiating that. Now, from a tribal perspective, a lot of the regulatory response to 6-PPD quinone has centered not only on the ecological impacts, but the tribes have been very central in driving the EPA to start looking at regulating contaminants like 6-PPD, which is a very common additive in tire rubber that keeps the rubber from degrading. That keeps tires from weathering very quickly and allows them to have the longevity that they do today. That 6-PPD is put in the tire to react with oxygen and ozone, which are in our atmosphere, that normally leads to the weathering of rubber materials. So that the chemicals that we use have a function that create the products that we use today. But we don't always know when they're released to the environment through all of the tire tread rubber particles that end up in our urban streams, or even the smaller particles that go into the air that we breathe, what the toxic effects might be. So here I wanted to highlight too that it's not just an endpoint of coho or an ecological risk. We're also talking about impacts to tribal communities. The fact that these coho salmon populations near these urban streams, like the Salish Sea, are not going to be able to recover unless we're able to find an alternative to 6-PPD that's less toxic. And also that there are ongoing impacts on other tribes that have already lost their salmon populations in the state of California. So being able to bring those back would be very important and very meaningful. Uh, and also another question, not just for coho salmon, but what other species are at risk? Are there closely related species such as Chinook salmon or sockeye salmon that have similar sensitivities to coho? And so those are the questions that our lab started looking at early on. What we found very interestingly is that coho salmon are the most sensitive of any species that we've tested, and some very closely related species don't seem to be acutely sensitive. We don't see the same effects, such as in chum salmon that spawn in the same streams that coho salmon do around the same time of year. They seem to be successfully recovering in a lot of those streams that have been restored for salmon habitat. There is some sensitivity in Chinook salmon, but again, usually at concentrations that we wouldn't normally see in the environment. But it raises the question of potential sublethal effects or things that can cause a delayed mortality that we're still trying to understand. So here is a list of some other salmonid species, such as rainbow trout and coastal cutthroat trout that we're finding that also are important species to our ecosystems and to our tribal communities in the Puget Sound. And they all have noticed in the literature, in the toxicity literature, that they exhibit very similar symptoms of that surfacing and that loss of equilibrium, and perhaps the same pathways of toxicity that occur in coho also are occurring in these species. Now, my current work, having wrapped up my laboratory studies and helped drive forward some of our understanding on those pathways of toxicity for coho salmon, was to come to the Confederated Tribes of the Umatilla Indian Reservation, where I've been working for the last year and a half to try and help this particular region on the Columbia Plateau, where salmon is also a very important first food species, and to learn about the role of first foods in the community, not only from a perspective of ecological health, but the cultural health, the importance to all of the gathering activities that is the center of the community's life here, and to really advocate for the idea of our regulatory systems measuring not just human health as a physical impact or ecotoxicity is just an impact on our ecological species, but looking at these as an integrated system of ecological and cultural health or ecocultural health. Now, the CTYR has a wonderful Department of Natural Resources and First Foods Policy program that puts out a lot of wonderful publications describing the importance of these first foods and their activities to try and educate other agencies and the public about how important these first foods are to tribal governance as a part of that co-management framework that is established by the treaty agreements. So this is a message that we're continuing to do in our program, which is Energy and Environmental Sciences, which is part of our Department of Natural Resources. And our DNR has a very clear mission that dictates all of the work that we do across our programs for fisheries and wildlife and water resources. And that is explicitly to protect, restore, and enhance the first foods, which are described in these main categories called the serving order, or the order of the foods that are served in the longhouse, which is tied to the creation story of the Cayuse, Walla Walla, and Umatilla people. And that serving order, which is water, salmon, deer, chauch, and huckleberry, for the perpetual cultural, economic, and sovereign benefit of the C2IR. And they also state that we will accomplish this by using both traditional ecological and cultural knowledge and science to inform the population and habitat management goals and natural resource policies and regulatory mechanisms. So on the left, you can see geographically where the Aboriginal title lands are that were established by the Treaty of 1855. Now, these were the lands that were ceded to the United States government. And through that treaty, the tribe secured the right to continue to fish and hunt and dig roots and pick berries across those ceded lands. Now, those boundaries that you see in the smaller inset map, those were the US government's interpretation of the exclusive ownership that they acknowledged for the Cayuse, Walla Walla, and Umatilla people. But that's not what would be the interpretation by the people themselves. So in the treaty agreements, there's always two versions of that history and two sides of that negotiation. In the larger map, you're seeing really what the tribe and the community recognizes as their traditional use areas. These overlapped with other tribes in the region. They shared these resources, they shared management of these habitats, and that continues today in how the community gathers and practices and acknowledges their relationships to these first foods. So I love this figure. The quote here is from that publication called The Gatherers Project. There's a copy of it that's available on the C2IR website that you can download and read more about the harvesting of especially the roots across those traditional use areas. Where it says first foods serve a fundamental role in the health, well-being, and cultural identity of the tribes. Now, the roots are the beginning of the seasonal round. Root Feast is the first feast where the women gather and have very important role in initiating the harvest season starting around February. So the seasonal role is just showing this phenology of care where the community begins and celebrates the latit or the Indian celery as it's otherwise known, where it first comes out of the ground at the end of winter and very early spring. And throughout the year, there is always some food to be gathered, to be harvested. And there's, of course, a lot of traditional tools. A lot goes into everyone having a role in the community as a part of the gathering and the sharing of these foods and the management of these habitats. And it's very important that when we talk about treaty rights, often fish get centered as the center of discussion, but we're always bringing it back to there are hundreds of species of plants, many species of fish, many types of game and birds that are just as important to protect that they are maintained at a quality that they're healthy for tribal members to eat, as well as accessible to be able to go gather. Now, the Department of Natural Resources uses this serving order as a framework for defining the restoration goals for the Umatilla River. So they've put out also the Umatilla River Vision, which is a publication written from this cultural resources standpoint with hydrologists and fisheries biologists and habitat managers to lay out the scientific and cultural metrics and describing what restoration means to the tribe. So they have very defined touchstones that go into all of the interconnections between the land and the water to be able to define those restoration goals. And a big need is to understand the impacts of the chemicals, which are mostly invisible and very difficult to detect without the analytical equipment, without the resources to be able to characterize all of the complex interaction of human activities that have changed these landscapes. So often restoration focuses a lot on important physical characteristics that are necessary for salmon recovery, like temperature, like having in-stream flows, controlling nutrient runoff. But there are many more contaminants, as you know, the research that I bring from WSU can help identify new interactions with both urban runoff, urban stormwater runoff, which does impact this watershed, but also agriculture is a predominant land use in Northeast Oregon. We have also other mining activities and lots of other sources of toxics to consider. So a first foods quality program has been an identified need for the tribe for many years. So the tribe has laid out a comprehensive plan from economic development to cultural resources to natural resources. And at least for the last 10 years, the tribe has been really focusing on trying to develop this first foods quality monitoring program to have the capacity to look at the integrity of these first foods and to protect their habitats. So this is the main goal that I was hired to come down and help lead the development of a toxics inventory and a strategic plan to look at how we can develop a monitoring program, what partnerships that we can make between our state and federal and local partners to help figure out what are the main drivers of limitations to salmon habitat and other first foods in the region. So as a first glimpse of our toxics inventory, I wanted to just share this map that shows you on the left some identified sites and sources, a lot of places where there have been contamination that have been mapped and existing data that's housed by the Oregon Department of Environmental Quality. We have many, many places across the landscape where contamination was known to exist, and likely there's a lot of residual contamination to consider, from cleanup sites to pipelines to the Union Pacific Railroad that follows much of the Umatilla River, all the way up into the uplands, which have historic mining and a lot of places that we may still have impacts from legacy pollutants like DDT, which is now a banned chemical. But for years, it was used to control bark beetle infestations in the Blue Mountains. And this is really important critical habitat for spawning and rearing for salmon. It's also where the roots are dug and where tribal harvesters are going that may be exposed to those type of contaminants that are still around in the sediments, and also where the huckleberries grow in the upland habitat. Now, these are not usually species that we have a lot of toxics monitoring data for. So our work can be really unique in helping to fill a lot of these data gaps to understand how the legacy contaminants or contaminants that we know are highly toxic but banned are still around, and layering those with what we're learning about emerging contaminants like 6-PPD quinone or PFAS substances or other metals that could be from the mining activity that historically occurred quite widely in the Blue Mountains. So from this, the connection between the monitoring data and the research to how we, as a society, are able to steer resources towards cleanup all comes down to how we frame our risk assessments. So the toxics inventory is just one of those inputs for us to understand what those contaminant properties are, where they are. There might be some information on how they're impacting human health or ecosystem health. But often the tribal framework is left out, or there's a lot of data gaps that don't allow risk assessments to really serve tribal communities and be protective of them to be able to uphold those treaty obligations, or the lack of information or lack of inclusion of existing data really creates a lot of issues for tribal members to continue to safely gather and restore a lot of access to their first foods. So a lot of the learning that I am doing as a tribal scientist is really understanding the intersection between the policy side and the Western science side, and combining that with the community's concerns, the community's activities. So a lot of our work in our program crosses a lot of boundaries. It really challenges us to have to develop new methodologies to make environmental risk assessments better serve our tribal communities and better serve the the threshold, or define those metrics so that the risks to the tribal harvesters are just as protective as non-tribal people. Now, the challenge here is that we have very little toxicity data to be able to even characterize what that risk means, even if we have the monitoring data. So this is well recognized in a lot of our current literature, that as we increase our analytical capabilities to measure and identify new chemicals like 6-PPD-quinone, the more we understand how little we really are capable of predicting their toxicity, or knowing how to manage these landscapes, given the number of chemicals that we have in our environments today. So as an example, EPA has identified about 297,000 chemicals of concern, but less than 1% of those chemicals have enough data to be able to do or to characterize a risk assessment where we could establish thresholds that can give us an idea of measures that could be safe, measures of safety that could protect either an ecological receptor or our human health. But here in the Umatilla, we have very little monitoring data to even be able to start a risk assessment process. So that is a major challenge. One of the few studies that have been done within the last decade were to look at larval lamprey tissues, lamprey being a really important first food to the tribe. But decades ago, it was considered a trash fish, according to the state managers. So the lamprey were intentionally eradicated by the state of Oregon by adding rotenone, which is a poison for fish, through the entire river to wipe out the lamprey. The tribe, since they've gained recognitions of their treaty rights and became co-managers, brought back the lamprey to the Umatilla. They seem to be doing well and thriving. And this study was looking at measuring contaminants in those larval lamprey throughout the Umatilla. And sure enough, we're finding those legacy contaminants like the DDTs, as well as flame retardants that are showing up in those tissues. And in the sediments where the lamprey are living, where they can burrow in the ground for up to 14 years, they have a very complex life cycle. We see quite even more contaminants of concern, both emerging and contaminants that we know are quite toxic to fish. Now, other types of first foods have even less of a success story in the tribe's restoration efforts. So the freshwater mussels are one example where they aren't coming back to the Umatilla River in many places that they've tried to transplant them. So there are many needs to understand how to clean up the contaminants that are entering the river to bring back many of the species, such as the freshwater mussels. On the other side of the more pollution tolerant species, while they are celebrated in the community and are brought back to the feast tables and brought back to the tables of many of the tribal members, those species also can accumulate contaminants that can be harmful to the tribal members who are eating them. So the Oregon and Washington Health Authority did a study on the levels of PCBs and DDTs and mercury, other types of known contaminants in fish, and issued an advisory that they were accumulating at a level that could be harmful to especially pregnant women and babies that are most at the highest risk. And from a public health perspective, educating the public about these consumption advisories to limit their intake is considered adequate to break those exposure pathways to protect the public's health. But these are damages to the cultural resources of the tribe. The recognition of their tribal treaty rights to be able to continue to harvest and fish also means that those fish should be healthy enough for the tribes to consume at desired levels. So, to continue to educate our public health co-managers on the need to address these cultural impacts is also another part of the work that we do. We also highlight the numbers of fish or the quantities of fish for our tribal gatherers greatly exceeds that of the general population. So most of those risk assessments that develop those thresholds of what's the safe level of fish to eat when those contaminants are found in those tissues usually assumes a consumption rate of about 17.5 grams per day. That's the general US population. But in the surveys of fish consumption done for the C2IR and other tribes in our area, they can consume up to hundreds, 540 grams per day or up above 1,000 grams per day, because salmon and fish were the mainstay of the diet here. They were the source for feeding the land and the people, and they were highly consumed. And that is just one of the first food species that we have data to be able to advocate for stricter water quality standards through our existing laws like the Clean Water Act, that we can use the tribal harvesting and consumption data to be able to advocate for greater environmental protections. But again, we lack an understanding on other exposure routes that are just as important through the roots and the other medicinal plants that are consumed and the berries. So there's still much more work to do to be able to really understand the tribal exposure route, looking at it through a traditional lifestyle. So for our work in our toxic strategic planning, our goal is to approach this problem that is very complex in trying to integrate both the cultural and the ecological and the human health risk assessment framework based on the tribe's own developed tribal exposure scenario, which very few tribes have a developed scenario that describes all of the activities that are important to conduct a risk assessment, and that work came out of their co-management at the Hanford nuclear site, and it's very well developed to be able to steer cleanup at this highly contaminated site so that the tribe can in the future return to gathering and exercising their treaty rights, which we still have a long ways to go. But using this scenario, we can apply it to non-Hanford sites, looking at other contaminated areas where we have even less information and less resources to understand what the contaminants are and what those risks might be. So we have to use this framework of adaptive monitoring or adaptive management, which forms that core in the center of asking our questions, monitoring, interpreting, sharing, that feeds into the next cycle of our adaptive management. And the boundaries, the practical boundaries of how we do this, depends on tribal policies and the intersection with US policies that limit what the tribes can do, but the tribes have a lot of sovereignty and a lot of ability to set their own policies and their own standards. And so the research that we do as toxicologists is very important to the establishment of those tribal policies. And so the core of our work at the center is really using our best available Western scientific tools and to combine those with the cultural knowledge, with the local knowledge, to refine our research questions. And that's what drives all of the tribes' work for restoration and their policy adjustments that are needed as we gain more insight on how to make clearly defined, tractable goals and the progress that we can make using our existing partnerships. So I wanted to give you one example of some of the goals and things that we have popped up during our strategic planning process. So one application of future monitoring is to look at known contaminated areas like dump sites where there have been a lot of historic dumping of vehicles to electronic to residential household garbage. Now, this particular site is located right along the Umatilla River in a really important habitat for salmon spawning and rearing. So there's a high level of concern about contaminants that could be leaching from this site into the river, probably very complex in terms of the chemicals that could be going into the river and have been for some time. Now, the tribe has to pay for the cleanup themselves. So these are one of the challenges of working in tribal government, where you have this mixed ownership status of the tribes having been broken up from their original trust land or land that was owned by the tribe itself. Through an act called the Dawes Act in the late 1800s. Those were broken up into individual allotments, where today there are tribally owned lands that are privately owned by individual tribal members. But the tribe can set a policy to come in and help clean up some of these areas. But because of that ownership status, they're very limited in being able to access funds like through the EPA, their brownfield program, that could be a significant source of helping these types of efforts. So without access to those brownfield funds through the EPA, we still have issues with these dump sites around the reservation that need to be addressed. So one of our goals is trying to figure out how to better work with the state and the EPA to leverage available funding to clean up these sites. Now, oftentimes in cleanup, cleanup can be very focused on known contaminants, and coming from a background where there's more we're learning every day about the unknown contaminants that could be driving toxicity to the emerging contaminants. The best way to be able to understand whether or not cleanup is sufficient is to look at your biological indicators. So one of the best biological indicators in an aquatic system is looking at the aquatic bugs, looking at the macroinvertebrates. So this summer, Oregon's Department of Environmental Quality did a training with myself and our toxics interns, and we got to go out and learn how to do the field sampling and learn more about the bugs that are living just downstream of this dump site. And the goal here in the future is to be able to leverage funding for doing our toxics research and being able to use these biological indicators to better understand whether we've achieved the cleanup goals that could be supportive for salmon by improving the quality of the food that they need to grow healthy and to be able to survive to finish their life cycle. So, but this is just one contaminated site among many that are very challenging. That the ultimate goal is to address cleanup to be not only to able to better understand how to improve these aquatic habitats and protect those biological indicators of health, but also to really highlight the focus of the need for research on the first food species themselves. So on not just a laboratory animal that is used as a representative animal for all fish species, which often times are not, they don't give us the information we need to understand when first food species are much more sensitive. Uh, so we need to be able to approach our cleanup to incorporate more of these broader questions of the habitat complexity and impacts on first food species in particular. Again, the mixed ownership status here on the map on the right is sort of showing that checkerboarding pattern where you have the outlines of the reservation boundary, and about half of it is owned in trust land by the tribe. So depending on where those cleanup sites are on the reservation, they may or may not be able to leverage existing funding for cleanup, like through the EPA's Brownfield program. And then around the reservation, we have many known sources of contaminants that can reach the river and a lot of those gathering areas. So some of them are shown on the map on the left. We have the nuclear reservation site, which is at the confluence of the Snake River and the Columbia River on the upper left. And so the tribe has been very involved in developing their exposure scenarios and cleanup efforts at that particular site. But also, just to the south of that, there's also the Umatilla Army Depot site where a lot of groundwater contamination exists. So the history of that particular site was that was an area where all the U.S. stockpile of chemical weapons were located here and then incinerated to be able to dispose of them. And through that incineration process, there was a lot of contaminants that were released into that site and continues to be an ongoing area of cleanup. There's also decommissioned coal plants in that area near Boardman and the Boardman bombing range, where we have a lot of military activity there and a lot of port industrial sites in that area. So it's a very challenging area from a cleanup perspective. And most of the gathering activities and most of the salmon habitat that the tribe is focusing on protecting is in the middle of the Umatilla Basin and into the uplands, where again, we still have many sources of contaminants, including the railroad and I-84 that has a lot of car crashes, a lot of diesel spills, a lot of ongoing inputs into these systems. So in order to leverage the development of tribal policy and using existing US regulatory policies, there's many intersecting agencies through the EPA or the Center for Disease Control, the Agency for Toxic Substances and Disease Registry, which can come in and help provide some information where there are areas of high contaminants that may be a threat to public health. We also have the Food and Drug Administration that has their own regulatory system for looking at how to set directives for investigating toxicity or the toxicological data that's needed to approve food and drugs, which is very different from a framework from the EPA. There are many laws that define the exposure limits that allow the tribe to be able to leverage this existing regulatory system to set higher standards to force industries or dischargers or emitters to have greater levels of cleanup. This is through the Clean Air Act or the Clean Water Act or the Safe Drinking Water Act. So there's many sides of policy to work on and from a tribal program perspective. But the goal here is to try to create those connections between the chemistry and the biology and policy to create these levels of acceptable risk or unacceptable risk to better serve our tribal communities. And again, this is just showing pieces of legislation and the dates that they were enacted and the ways that these policies are connected to the various levels of developing our strategic plan and the policies that are needed. So there's a lot of the legal justification and a lot of the monitoring work that we do has to be able to be held up in court to be able to meet these standards that are defined by these existing laws. So when it comes to regulatory toxicology in practice, we know that in order to meet these standards, to have the quality of data that we need that drives the cleanup, that drives changes in these policies, it really relies a lot on the laboratory data that's generated, like our lab at WSU. And oftentimes those on the policy side don't always understand a lot of the assumptions or how simplified these models can be. And oftentimes they're not as representative to the real world conditions as we would like to be, especially for tribal communities that are impacted through all of the different types of exposure routes and all of the different types of needs that they have for understanding impacts on their traditional first foods. So as an example, oftentimes in the laboratory, our toxicology thresholds are driven by an understanding of mouse models or zebrafish, which is a fish that you can find at the local pet store. And when we are able to look at the thresholds that affect zebrafish and have that numeric concentration of that single chemical that was exposed to that fish, oftentimes that's used in place of representing a safe threshold for all fish species. But in reality, the way that we set those thresholds, which might be based on a zebrafish model or might be based on a mouse model, it misses the reality of these multiple chemical exposures and the need for a cumulative risk assessment, a need to understand that the data that we're generating from that single chemical and that laboratory model doesn't reflect the reality of the chemicals that we're exposed to. We have different sensitivities depending on our demographic backgrounds, our age, where we work, the places that we live, the types of foods that we consume, the types of products that we use. These are very complex, and it's not just the amount of stress from the chemicals that we're exposed to, but also through non-chemical stressors or all of the ways that we might have our health may be impacted for things that are not directly related to chemical exposure, but still set and still affect our sensitivity to those exposures. And from a tribal first foods perspective, we already know that species vary quite a bit in their sensitivities to chemicals. So even in the toxicology data that exists, we know that, that laboratory model in the lab, there is very likely to be another species if we kept, testing multiple species that will be more sensitive. So to be able to have existing data generated in our toxicology labs that focus on first foods will really help fill some of those data gaps so that we can use that information to drive safer limits and safer thresholds through our existing water quality and air quality laws. So the basis of being able to look at the sources of all of the chemicals that are entering our environment through the products that we use, the way that, industry and new chemicals are regulated through the EPA, we're coming to understand the limitation of our ability to manage them on a single chemical basis, on this idea that Western science has the ability to measure and predict and to understand from this managerial or this conceptual model, and to have that be the basis of our regulatory system is very limited. And we know that the way that our regulatory system works is not equally protective of all species, of all people. So there is a need to shift from the model that centers, what usually in a risk assessment framework has a white male, typical general American diet scenario, to looking at from an Indigenous perspective, from the perspective that has been advocated for C2IR's First Foods mission, is that we live in a democracy of species. We know that humans are not centered when it comes to ecosystem health, that all fish and water and species are interconnected in how we define health and how humans can live sustainably, and how the tribes have lived for thousands of years. And that will continue to be the framework by which C2IR generates their policies and generate and conducts our research. So from a standpoint of what's needed as a path forward to co-manage the chemical habitat to be protective of first foods, the most important part of that mission is to train future tribal scientists. And that has been a major focus of the work that we do in our energy and environmental sciences program, is to work with our youth from high school on through undergraduate, through their graduate degrees, so that we can train them with the technical expertise, but they come with their lived experience from the community. They come with the assumption that their first foods gathering knowledge and their experience is incredibly invaluable to their work as tribal scientists, that can't be trained. It has to come from the community itself. So while we can hire wonderful scientists to staff our research, our DNR programs, the goal is always to create these educational pathways to support the students in their connections to their communities and first foods throughout that entire career development training process, whether they want to be technicians or they want to be future scientists. And this are just a couple of pictures from this summer where you can see our air quality technician in the upper left giving a tour for our monitoring station to our tribal youth. We went out and did a lichen bioindicator project with them, characterizing some metals that were taken up from air in around that site that's owned by the tribe. They got to learn about their tribal government and talk to their board of trustees and understand how important that their voice is in the tribe's leadership and the development of policy. And then we also developed a teaching game for the younger students that is based on a board game called Pandemic. And we turned it into a Umatilla River vision game where we used the place names atlas that has the Umatilla, Cayuse, and Walla Walla terms across the Umatilla Basin, so the students can play the game and understand elements of DNR's First Foods mission and work collaboratively in the game to address wildfire risk and air toxics. So there's lots of ways that we are working to help restore the Umatilla River according to DNR's First Foods mission and goal. That's not just doing scientific research. It's really based in community. It's based in centering tribal knowledge in language, learning how to think outside of our traditional Western science way of of managing our habitats. While it can be very valuable, um, it's really that reflection on what existed before 1855. That's the standard that the tribe sets for what needs to be restored. What exists today? What tools do we have? What partnerships do we have? And together, that combination of, Western science, traditional knowledge, really reframing our way of thinking about our role in our ecosystems, our role in community, that is central to understanding how to define health and how health can be restored. So on this last picture, I wanted to have an aerial photograph of the Columbia River, where you can really see the impacts of all of the development since 1855. It's heavily industrial, agriculturally impacted. The hydropower development throughout the Columbia has significantly changed this system. But what remains through all of that is an understanding of how clean and cold the water was. That's still something that the elders talk about every day, that they remember a time when they could drink out of the water in these places. They remember the sacrifice that the salmon made for the people from the creation stories. And they will always be the center of understanding how health can be restored in the Columbia River Basin today. So that is important, not just for tribal people, but for all of us to reflect on that language of the treaties about the obligation to the first foods, to protect those rights to fish and hunt and gather roots and berries, according to the management frameworks defined by the tribes themselves. But non-tribal people, we will also benefit from that by better understanding what's needed to place our infrastructures in a way that can be able to address some of these complex challenges that we have, from stormwater to agricultural runoff to redefining what those safe levels can be for substituting the most harmful chemicals that we have entering our environments, chemicals like 6-PPD. And tribes are central to defining all of the methodologies that are needed to accomplish that. The final slide here, I wanted to say, on a personal level, as an Ojibwe woman, a person who's been a part of many different Native communities, I would say that connecting to our language, to our families, to understanding how we came to be in the places that we live, that there's always a need to connect, to slow down, to spend time with our traditional foods, no matter where that they may come from. That to me personally has been the center of being able to do this work, of connecting with our foods and being able to bring them to the places that I live in Puyallup, Washington, and now down here in Northeast Oregon. And we all have the foods that sustain us. We all have something to be grateful for, to really let us know how important our foods are to our way of being. And they help teach us what we need to reframe our way of thinking, to be able to approach the work that we do with integrity and with honesty and with love. So with that, I'd be happy to answer any questions.
Okay, thank you so much. This was the most wonderful presentation. Can you hear me?
Yeah, I can hear
Okay,
you.
good, good. A wonderful, wonderful presentation. And let's see, we can turn it over for a few minutes to Diana. I know this has gone over a little bit in time, but for those of you that are still on, um, you can enjoy hearing some answers to questions. So I'll turn it back to Diana now. And she may have some questions for you.
Course. I don't know that I have questions, just incredible appreciation. I just want to say I really loved the holistic, comprehensive way that you talked about this and pulling in the toxicology, the indigenous ways of knowing, how you're integrating youth and all of this was just a really way to show the importance of the work that's being done. Uh, so I'll let folks take a chance, put your things in, or put your questions in the Q&A, please. We did have one question early on, so I've been holding on to that. I'll read it out for you. But the question was, can you recommend methodologies for multiple partners to gather information on levels of 6-ppPDQ and how we can bring together transportation planners, road maintenance, natural resource scientists to implement improvements to mitigate this chemical? And they said, other than bringing back old tire manufacturing that doesn't contain this chemical, are there other strategies for mitigation?
Yeah, well, a lot of the work that we had so at WSUA we had the Salmon Toxicology Lab that helped us identify 6-ppd quinone. We also had a low impact development lab, which is also called Green Stormwater Infrastructure, where we found many different types of treatment using amendments like soil and compost that is very effective at removing 6-ppd quinone. So we know we have infrastructures that can help prevent it from entering our urban streams. The problem is implementing them at scale. So we can place these at point sources, but we don't have an answer yet about how to have them remove all inputs of 6-ppd quinone to our urban stormwater. And then also it's important to remember that 6-ppd quinone can travel through the air. So stormwater treatment is not always going to be the answer to chemicals that are coming from our transportation sector. So the answer to 6-ppd quinone, I think the consensus is on replacing 6-ppd with an alternative that is less toxic. That's always the best answer when it comes to harmful contaminants. Is source control, is replacing that chemical with something else. So that is an active area of research to find an alternative to 6-ppd. There are some studies that are out there that are available that have looked at that. So I'm hopeful that that will be on the near horizon. But even if we found a replacement today, it will still take decades in order for it to actually be replaced in tires on the roadway. So, a lot of the salmon populations that are at risk, they're declining very rapidly. So, it's not always feasible to think that we can understand on a chemical by chemical basis, identifying the chemical, doing the research that's needed to find a replacement. We really need to be rethinking about the amount of chemicals that we're producing as a society, how many new chemicals, hundreds of new chemicals every year, and really start thinking about our relationship to our built environment in a more sustainable way. Uh, and tribes are really at the center of that conversation because their health and their resources are the most at risk. So the National Tribal Toxics Council has for years sort of developed a tribally informed framework to screen new chemicals. That got very close to being adopted by the EPA this year, but unfortunately, that is no longer going to be an emphasis, but it will continue to be something that tribes push for through groups in the EPA to center tribal exposure as that group that is often most at risk, so that the way that we screen our new chemicals is more protective of our tribal communities. That will be one way that hopefully we can prevent harmful contaminants like 6-ppd quinone becoming a problem in the future. But again, there's there's going to be lots of pieces that are going to need to be addressed to make these changes at a societal level.
Thank you. And then related in a sense to that, but there was a question asking if there are studies looking at soil contaminants in areas where root resources are being collected.
Yeah, that would be a very good way for us to look at the soil, look at what's being taken up into the plants. Um, I know that a lot of the places that are roots are gathered are on both public and private land. So having access to go out and doing the sampling is not always available. But starting with what's the proximity to likely sources is where we're trying to develop an initial sampling plan for a lot of the roots and areas that are being actively utilized by the community. But absolutely, starting with the sediment, and often it can be quite difficult at looking at the plant material itself. Um, it requires, having dedicated labs to be able to do that research and ask those questions. And just hopefully by promoting this work, that more research labs can start directing their focus in that area.
Thank you. And since you mentioned sediment, that leads directly into the next question, where they ask, do you have any ideas about how to remediate things like the toxins in the bottoms of waterways, so sediments, but without releasing them where they could do even more damage? Related to that, could you think about climate-driven increases in stream water temperatures, the lower Snake River dams? So sort of a lot there, but thinking about toxicants in sediment and how to remove them, as well as other things like stream water temperatures and dams that might be contributing to release of toxicants.
Yeah, so the restoration side is not my area of expertise, but I know that there are a lot of concerns that if you were to directly go ahead and dredge and remove a lot of that contaminated sediments, you have to consider resuspending a lot of that. And the toxicants that are associated with them. So it's not always an easy fix to go and dig it out and haul it away. And then where do you haul it away to? Like that has to go somewhere. So it'll be shipped to a landfill that can accept that hazardous waste material. So um, again, like it's always very challenging when we think about how to address these most contaminated areas. That requires a lot of a very thoughtful approach and a lot of care. So having tribes be involved in that usually drives a little bit more slower process and a lot more careful process, which is necessary.
But also a more thorough process.
Yeah.
Um, Next question. Could you comment on any research you are pursuing that might be addressing population growth and its impact on ecosystems?
Human population growth. Yeah, I think that's a big challenge for urban planning is how do we decide where people live in a way that's going to be sustainable? So our research has looked at, transportation and just people, the amount of cars driving down the road is really one of the major problems that need to be addressed. So we definitely could be incorporating more public transportation, ways to reduce the amount of vehicles on the road, that would be a major benefit. That would improve a lot of the quality of our ecosystems, especially these urban streams. Where we decide to place those infrastructures is important. That's an ongoing question of do you start with the most degraded habitats and start implementing green stormwater infrastructure there, or do you start in the places where you see development is probably going to be expected in the future and place those infrastructures there to be able to prevent degradation of that high quality habitat. Again, this requires a lot of public input in order for these decisions to be made. So the more that the public really understands the complex issues involved in a lot of the chemicals in question, I think will also be beneficial to handling a lot of that urban and population growth.
Thank you. We have one more question in the Q&A if you're up for it. Uh, they're asking if you could recommend some models for adjusting policies based on modern Western legislation with policies that align with Indigenous knowledge and experience.
Yeah, well, I talked to the National Tribal Toxics Council. They write policy letters all the time from the tribal perspective, and absolutely, that's a big focus and a big conversation. The challenge, I think, is that a lot of the policies involved with chemicals management is highly technical, and usually very few people can sit down and read through these policy changes and understand how to make recommendations from an Indigenous framework. And then a lot of the assumptions that tribal communities are really saying are not a part of the framework to begin with in the policies that are being adjusted or changed. So yeah, I think that really speaks to the need of having more tribal scientists, being able to start with that understanding that comes from their lived experiences and get them into some of those technical fields where then they can really understand how to generate leverage on policy reform.
Thank you so much. I know it is hard for you to have to talk and read the chat, so I just wanted to convey several people put in the chat and in the Q&A just their appreciation for the talk and how they've learned quite a bit about this. So a lot of compliments from the whole audience.
Great. Yeah. Well, thank you so much.
Yeah, and thank you, Diana, for monitoring the questions. There's a whole lot of chats coming in all of a sudden. You can probably see them.
Yeah.
Uh, and this was a wonderful presentation. I think it was so in-depth and so important. And I want to tell all of our audience that this will have been completely recorded and it will be available on our website, which is Indigenous Education, indigenousedu.org. And you'll be able to download it for free and use it in your classrooms or whatever you want to do with it. So it'll be available to you. All of our in this whole series of 26 lectures have had that. I also wanted to say, I wanted to thank Chris Tarrant of Tarrant Solutions in Friday Harbor, who is our IT expert, because this was the most challenging one we've ever had for some reason. I don't know why. But I think it could be because I was coming from a hotel in Alamosa and my Wi-Fi kept going on and off. But anyway, we call this the coyote factor when this is a Navajo thing, but when something goes wrong, we think of it as a coyote coming in and trying to disrupt everything. And he really tried his best today, but thanks to a lot of Chris's good work, he didn't succeed, and we were able to continue. I also wanted to say a thank you to Ashley Tarrant, who developed our beautiful poster to advertise this session. And I just want to thank you all for being here. This was a most informative, just wonderful presentation. Thank you so much for taking the time to share with us. And I hope you all have a good weekend. So from all of us, thanks to everybody.